Welcome, students, to March of Dimes High School Science Week. My name is Madison Dodds, and I am a member of the Salk Institute's Education Outreach team. This event is a Salk tradition, and this is the 31st annual event. We wish we could have you on campus with us, but we're proud to continue this event in a virtual space. This is an event that not only our team looks forward to each and every year, but our Salk researchers really look forward to interacting with students and having them in the lab. So they are very eager to interact with you throughout the week and answer your questions. So for those that have not had the opportunity to visit Salk, or if this might be the first time you've attended a Salk event, just a little bit of background. So Salk is not an acronym, but it was named for Dr. Jonas Salk. You may have heard his name recently in the news, as he was a key figure in another public health crisis, and that was the polio epidemic of the 1940s and 50s. He was the first developer of a safe and effective polio vaccination. He then went on to pursue another dream, which was to create a collaborative environment where researchers explore the basic principles of life. 60 years later, the Salk Institute is a world-renowned basic biological research facility. We're located in La Jolla, California, right across the street from the University of California, San Diego, and it's a beautiful place to work. Just as if you were joining us on campus, we would be welcoming you into the courtyard, and that's shown in this image here. So Dr. Salk worked with famed architect Louis Kahn to design the Institute. If you're interested in architecture, there is a virtual tour on our website. The fun facts about the Institute that we're going to be highlighting throughout the week. So you might notice this fountain that runs down the center of the courtyard. And on the equinoxes, both in the spring and the fall, it will align perfectly with the sunset. And off in the distance is the Pacific Ocean. It is a beautiful and inspiring place to work. But probably my favorite thing about the courtyard is this quote in the front. It's kind of hard to read, so I'll read it to you. It's right here. So Dr. Salk said, "Hope lies in dreams, in imagination, and in the courage of those that dare to make dreams into reality." Throughout the week, you're going to get to meet some of these individuals who are daring to make dreams into reality. So a quick rundown of what to expect for the virtual lab tour today. So we'll start with a quick introduction and some basic information about some other programs that Salk Education Outreach has for high school students. Then we will air our virtual lab tour video and will be followed by a panel discussion. So each lab tour is a standalone experience, so you don't need to watch one to understand the next. You're welcome to attend only one or a couple that align with your interest or all of the events. We encourage you to check out at least a couple because you might be intrigued by a field you've never considered before. So each lab tour is a separate link, so if you're planning on attending more than one event in a day, please make sure to use the individual link to access the lab tours. While we are so hopeful the event will run smoothly, we've all had technical issues arise. So if that does happen, please be patient as we do our best to resolve it as quickly as possible. If you do run into any technical issues during the program, please email education@salk.edu and we'll do our best to get to you as soon as we can. So a little bit about some of the EO programs that we have. Sorry to get in. We are so excited to interact with you throughout this week. And that's our favorite part of High School Science Day. So we're going to be using student questions to really guide our panel discussion. So to submit a question, you're going to go to the link that's found in the video description right below this. You're welcome to submit as many questions as you would like. We thank you in advance for considering it, for thinking a little bit deeply about science, and for being willing to ask questions and be brave. We'll do our best to get to as many as we can, but we are short on time, but we'll do our best to talk about a variety of different subjects. If you want to learn more about the lab's research, including information about our panelists, what you'll do is you'll click the Show More, and you'll have a link to the website that has the bios for the moderators and the panelists. You can learn a little bit more about them as individuals. You're welcome to submit questions using the form to an individual panelist or for everyone to consider. So now a little bit about some high school programs that we have if you're excited about science. So the first is the High-Toff-Brody High School Summer Scholars Program. So for this program, you must be at least 16 years old to participate, and it is a full-time program. So the program consists of, it's 8 weeks. The first week is a biotech boot camp, which is a crash course in a variety of different lab techniques that you might use, followed by 7 weeks in the lab paid, where you get to work side by side with a Salk scientist to conduct real life research as we contribute to some of these groundbreaking discoveries. You're also going to have the chance to develop some science communication skills that will help you in the future. But outside of that, we have a variety of different other opportunities that our interns participate in, like industry visits, seminars. We also like to have a lot of fun. So this picture up in the right-hand corner is our interns at Salk Picnic, which is a fun day where we get to go outside. All of the Salk employees, there's volleyball competitions and lunch. It's a lot of fun. So while we're hopeful the program will take place in 2021, it is dependent on the public health conditions at the time. So please check out our website in March for more information about the program status. Okay. But even if that program, we might have to go on hiatus again, we do have the Introduction to Research Science and Communication virtual program. So this is a 5-week professional development program for students, and this program is not limited to San Diego County students. The program is open to all U.S. residents enrolled in high school during the 2021 academic year. So another great thing about this program is it's mostly asynchronous, and it's a lot less of a time commitment for our students. It's only about 5 to 10 hours a week. The program elements were taken from the Hightoft-Brody High School Summer Scholars, so you're going to be building it towards a skill set that will help you in future internships. So the program elements include reading journal articles, analyzing data, preparing a scientific presentation, and virtual labs. You get to work towards a final capstone project on a topic of your choice, which is pretty much an independent research project. It's a lot of fun to get to hear all the things that students are curious about. To be considered for this program, you'll complete a opt-in form on our program lottery on our website. That's available and up right now. So we'll definitely make sure you have our contact information so you can opt into that if you're interested. The lottery will close March 31st. Again, I told you so much information about these exciting programs, and it might have sparked a lot of questions. So if you do have any questions, please email us at education@salk.edu or check out our website for more information. There's also an FAQ page for frequently asked questions that can help you out. So this event is not possible without the generosity of quite a few people. So we would like to thank the following sponsors. March of Dimes High School Science Day is made possible by the generous support of the Anne and Neil Blue High School Science Fund. Our special sincere gratitude to the Blue family and the supporters, partners, volunteers that make Salk education outreach possible, including the March of Dimes. So now it's one of our favorite times, and that's to get started on our virtual lab tour. So today's virtual lab tour will feature the Gene Expression Laboratory of Dr. Geoff Wahl. The videos that you're about to see will give you a glimpse of what a day in the life of a scientist is like. Each video was designed and filmed by the scientist. Each one is a little bit different. They will highlight the tools and technology that our scientists use, but also introduce you to people that work there and make these discoveries possible. Also, it'll give you a couple different career options if you're interested in pursuing a career in the STEM fields. In the opening presentation, you heard about the research that the Wahl Lab does, and now we get to highlight the incredible talent that brings these discoveries to life. And they're going to share with you some of those techniques that they use to make these discoveries. It is my pleasure to introduce the Gene Expressions Laboratory of Dr. Geoff Wahl.
Hello, welcome to the Wahl Lab. My name is Razia Naeem, and I'm a research technician here in the lab. One of the most important molecular imaging tools today, and one that we use in the lab, is GFP, green fluorescent protein. GFP was first isolated from the jellyfish Aquaria victoria and exhibits bright green fluorescence when exposed to light in the blue to ultraviolet range. Many colors have now been derived from the green fluorescent protein and red fluorescent protein that was initially derived from coral. One way to use this tool is to use GFP as a reporter gene. GFP can be linked to another gene that produces a protein that you would like to study. In our lab, we use transgenic mice that contain GFP linked to a gene of interest, in our case, SOX10. This allows us to track protein expression. The GFP allows us to track when SOX10 is transcribed and translated by making SOX10 expressing cells green.
Hello everyone, my name is Vikas and today I will be explaining how and why we genotype. Say we inserted a gene for a fluorescent protein into a mouse's genome and breed it with another mouse. How can we tell whether the children's DNA has that gene of interest? To do this, we use a technique called polymerase chain reaction, or PCR, which allows us to amplify short, specific regions of the mouse's DNA by millions of times. We can set this up so that the presence or length of these segments can tell us whether an inserted gene is either present or absent, seen by the pink arrows. The large amounts of DNA can be visualized by staining, but more on that later. PCR works by first denaturing the DNA, or separating the two strands from each other by heat, as seen in the red box. Very small sequences of nucleotides called primers bind to highly specific regions of the DNA strand, and the binding site of these primers determine the region and the length of the segments amplified, as seen in the yellow block. Then polymerase enzymes add free nucleotides from the solution to build complementary strands of DNA, as seen in the orange block and the figure on the right.
Hi, my name is Yasol. I work at Gene Expression Laboratory, Wahl Lab here. And today I'll be showing you how to isolate DNA from animal tissue and use them to perform polymerase chain reactions to amplify the gene that we're looking for.
The first step of genotyping requires an extraction of DNA from our mouse tissue. To do so, we add a lysis buffer that has a detergent to break down lipid layer of the membranes to release the DNA from cells, as well as proteinase K, which is an enzyme that removes proteins that degrade the DNA. This process requires an incubation at 55 degrees overnight. Afterwards, we isolate out DNA using alcohol because DNA is insoluble in the presence of alcohol. Adding isopropanol to the lyse buffer mixture will precipitate out the DNA, as you can see here. Then we clean the DNA precipitate with ethanol to remove any remaining impurities. After evaporating out the alcohol, we resuspend DNA in a buffer that dissolves and protects DNA from degradation. To perform PCR to amplify our gene of interest, we make a master mix with the buffer that provides a suitable chemical environment for the DNA polymerase activity, a nucleotide mixture called DNTPs providing building blocks for DNA synthesis, primers that flank the gene of interest, and a DNA polymerase that has high heat tolerance that can work well under extreme heating condition. Then we add our DNA samples we prepared to provide a source of template for the PCR reaction. This reaction mixture is then moved to a machine called thermocycler, which enables these samples to go through multiple cycles of heating and cooling to actively amplify the gene of interest.
Hi, my name is Cynthia Ramos, and today here at the Gene Expression Lab Wahl, I am going to show you how we visualize the DNA that comes from the PCR reaction by using the agarose gel electrophoresis method.
Agarose powder is dissolved in a buffer solution which serves as a matrix for DNA to flow. A standard is used to compare the sizes of DNA fragments in our samples and is loaded into one of the gels. Next, we pipette some loading buffer and mix it into our PCR reaction. The loading buffer contains a colored dye which allows us to track the DNA as it moves through the gel matrix. It also contains glycerol, which will make the sample heavy so that when we load our samples to the wells in the gel, the sample will sink in the bottom of the well. The tip of the pipette is carefully put into the well and each sample is loaded. The lid is attached and connected to a power supply. DNA is negatively charged, and so all DNA flow towards the positive electrode, and fragments will be separated according to size. Smaller fragments will move faster, and bigger fragments will move slower. The agarose gel contains ethidium bromide, which is a stain that binds to DNA. Once the gel is exposed to UV light, the DNA fragments will give a fluorescent signal, which will enable us to see how far the fragments have migrated through the gel matrix. The size of the fragments that were amplified in our PCR reaction will be compared to the known sizes of fragments in the standard ladder. The image of the gel is captured in a picture.
Hi, my name is Cynthia, and I am a senior research assistant in the Gene Expression Lab Wall. When I say senior, I mean that I have been working as a research assistant for over 40 years. My job has brought me to 3 different countries in 3 different continents, working in 3 different scientific fields of research. In the Wall Lab, I provide technical assistance to our postdocs and research scientists. I also train undergrads and new technicians, sometimes postdocs too. And at the same time, I also learn from these people. There's always new techniques to learn in the lab. And every day is a learning process, a learning experience. And that is what makes it exciting to be working in the lab. Now I will allow our panelists to introduce themselves. I will start with Yizol Go, who is a lab tech in the same lab.
Hi, thank you for the introduction, Cynthia. So, as Cynthia said, I am a lab technician at Gene Expression Laboratory Wall, and my job title is lab technician. So, most of my job includes supporting postdocs with their projects, and also Cynthia, who's our lab manager, taking and maintaining the lab so that it runs smoothly. So, and I started working after 1 year. After 1 year in working industry, and I moved here, and the experience has been so great. Everyone's really nice, and yeah, so it's a really fun job here. And next will be Nikki.
Hi, everybody. I'm Nikki Lytle, a postdoctoral fellow in Dr. Wahl's lab. I've been in the lab now for 2 years, and I work closely with Razia, Yesel, and Cynthia on many projects involving both pancreatic cancer and breast cancer. I'm originally from Oregon, but I moved down to San Diego about 8 years ago to first do my doctorate, complete my doctorate at University of California, San Diego. And then I just crossed the street to Dr. Wahl's lab here and continue working on aggressive metastatic cancer. So, Razia, please introduce yourself.
Hi there. I'm also a lab tech. My name is Razia Naeem, and like Yesel, I'm a lab technician here at Gene Expression Laboratory Wall. And I'll be working, it'll be 3 years since I started working here. And I also work, same as Yesel, where I work with postdocs and staff scientists to help conduct research.
So the first question is, what is the most stressful part of working in the gene expression lab? And what is the most rewarding aspect of it? Yizol, would you like to
answer that, or Razia, maybe?
I can go ahead and go first. So the first thing that just comes to my mind when it comes to most stressful part is whenever you do any research projects and learn how to do new techniques, whenever it doesn't work out, we have to go through something called troubleshooting, and it might take a really short time or a long time to figure out how this technique could work out so that we can continue our project. But the time it takes sometimes might be very long, and it could be very stressful. But at the same time, the most rewarding part of it is figuring out how that technique works out, and our project can be moved on. So I can say that that is one of the most stressful and rewarding part of it.
Second question is, what do you enjoy the most about working in the lab? Could we have Razia answer that question?
Well, what I enjoy the most about working in the lab is coming into work and learning something new. And I feel like I learn something new each and every day. So that's been the most enjoyable thing. And every day I work with just incredible people. And so that is also one of the things I most enjoy is the people I work with, what I learn. And
it looks like Cynthia is having a little bit of technical issues, so we get all that settled. Nikki, I believe that you were sharing a question about the stress and some of the most rewarding aspects. Would you mind continuing on that before we move on to another question?
Oh, yeah, sure. I was going to talk about how, despite being in research for, probably 15 years at this point, I still chronically underestimate how long experiments take, and it's impossible. Like, I think I have a question, and I design the experiment, which I really enjoy, that part of it, because there's a lot of creativity that goes into designing experiments to answer specific questions, and then you set it up, and you think you're going to have the answer in weeks, and then 2 years later, you still don't have the answer. So it's really like this. Science is an ever-evolving process. You really do your due diligence and trying to read as much as you can and talk to as many people as you can to come up with the best approach to answer a question. And then sometimes it doesn't work out the way you think it's going to. So there's a lot of restarting.
Razia or Yesol, can you relate to that about underestimating the amount of time it takes to do an experiment, or is that something unique to Nikki? What do you guys think?
Yeah, I totally agree with Nikki.
Yeah.
Because sometimes, we like Razia and I, we see her. We also support her in her projects and stuff. So we see her very closely what she means by it takes longer than she thinks. Yeah.
And how many times you say, "Oh, of course I'll be home by 7," and then it's like, "Oh, I was just kidding, it's 8," and then you're like, "I don't know, I have no idea when I'll be home."
Science does not follow your timelines. As we can plan our best, but science has its own timeline.
Yeah.
Awesome. So, another question a student had is, what would cause a cancer to become more aggressive? So I think, Nikki, you had done some research on this, and you had contributed to the project that Geoff had talked about in the opening project. Would you mind sharing what would cause a cancer to become more aggressive?
Of course, and Geoff really hit on a lot of the stuff that we our lab and others work on in trying to understand cancer evolution. So he had talked about intratumoral heterogeneity, so this idea that one tumor cell can be very different than another tumor cell within the same tumor. And our lab really works to try and understand that heterogeneity and how that heterogeneity can contribute to therapy resistance, which Geoff had talked about with Herceptin. But also one thing that we also look at is how possibly tumor cells can accumulate additional mutations that make them more or less aggressive, and those additional mutations can change the phenotype of the cell or the behavior of that cell. So one cell can accumulate mutations that allow them to leave the primary tumor and spread to distant sites, which we call metastasis. And Geoff had mentioned metastasis, and really metastasis is one of the major drivers of patients succumbing to this disease. So,
Yeah. Yesol or Razia, would either of you like to build on that, or we think Nikki nailed it?
I think Nikki nailed it.
Yeah, me
Yeah,
too.
that was very well put. I think it was. Thank you for sharing. All right. So let's have Razia. I have a question. So let's have you take this one first. Why did you choose this job, and what led you to pursue cancer research in particular?
So my father was diagnosed with pancreatic cancer. And so once, after that happened, I really wanted to work in the field. I wanted to, contribute so that nobody else has to, go through that. So that's what really got me into the lab. I started off as a volunteer here, and I've been a lab technician since.
Awesome. Thank you for sharing that. I think at the beginning they had the student question about if you know someone that's been impacted by cancer. So for you, your research is not only driving your curiosity, but you have some personal buy-in to that. So thank you for being a part of Salk. All right. Yesol, for you, would you like to follow up? I'd love to hear your perspective on what brought you to cancer research.
So I was actually studying biology as an undergrad, and I was always interested in what academic research would look like, and what really wanted to kind of see what that experience would be like. So that's how I applied and got in, and I just really am loving how interesting and like daily you learn, like Razi said in the beginning, like you learn new things every day. And that's just what research is always like. You find out new things that you were never expecting, and that's the fun part of it. Yeah.
Awesome. All right. We have another question. Have you had any experiments that went completely against your hypothesis? So let's hear from Nikki. Have you had any experiments that went against your hypothesis?
Yes, always. there's sometimes you think there's going to be an interesting difference, like you're testing two different situations, and you think that one situation may cause different cancer behavior, and there's absolutely no difference. Like convincingly, there was no impact on the cancer behavior from that. But actually, recently, my work has been shifting towards how the body interacts with the tumor. So, for example, pancreatic cancer patients that have forms of liver damage can often have higher rates of metastasis to the liver. So the hypothesis was that the liver damage will essentially help metastatic tumor cells in the liver survive and grow. And I'm testing a number of different liver damage paradigms. Some of them seem to be showing me that, yes, indeed, that liver damage is helping pancreatic cancer metastasis. But I had one form of liver damage that actually showed the reverse, that the damaged liver, the damaged liver itself did not promote metastasis, it actually blocked metastasis. So this is very interesting because now it really suggests that it's not just all liver damage, it's like different forms of liver damage may have differential impact.
All right, it looks like we have Cynthia joining us back. So Cynthia, since you've been at Salk for a number of years, let's have you take this question first. What has been your longest or most difficult project to support?
Well, I have been only 5 years at Salk. As I said, the 40 years is in between 3 countries. I did plant research, and then I was in a neurobiology lab, and now in cancer. So I'm still a baby in cancer. But speaking of techniques, as a research assistant, when I joined the group, I was introduced to a very specialized technique of dissecting very, very tiny, immature organs using the microscope and fine tools. So that was for me very challenging, but in the end, like we have good mentors here and practice, I was able to overcome that. And now I am proud to say that I am able to train other members of the lab. Probably, you know, when I retire, there will be people who will be continuing this technique.
Alrighty, so I will pass it back off to our moderator to continue asking questions. Thank you so much for letting me jump on in and hijack and get to ask a couple of the questions I was curious about.
Sorry about that. Thank you for jumping in, Madison. So we have a very interesting question here from Joshua Von Trapp, and he's asking, have any cancers been cured? Nikki, would you like to answer that?
Yeah, cure is a difficult word because there are certain cancers in which we've significantly improved overall survival. One classic example of this is chronic myeloid leukemia, or CML. And patients with CML used to have a very poor prognosis. They would unfortunately succumb to their disease in under a year. But then there was this wonderful discovery, actually partly due to some of the work out of another faculty member at Salk, Tony Hunter, where they had discovered an inhibitor of a receptor tyrosine kinase, which is a protein that CML depends on. So with the advent of an inhibitor of that protein, now patients can live 40 years with CML with no problem. The risk is that sometimes over time, the cancer will find a way to no longer depend on that protein and can eventually evolve to advance to a more aggressive, untreatable disease. So cure is hard. We hate to use the word cure. Like, we're striving for a cure, but we understand that there's always a risk of cancer finding a way to circumvent the therapies that we have.
Thank you for that very encouraging answer, Nikki. Do we have other questions?
I see an interesting question here that maybe Yesel or Rosie or Cynthia could even do, which is, what is a single piece of advice you would give to a high school student going to college?
Oh, for me, it's been a long while. but I think it's important that you have the interest, the curiosity, and the passion for something like research, cancer, as I said, neurobiology, or plant, in all of the different fields of science research, scientific research. I think for me, I didn't notice that time passed because every day I come to work and I am inspired and I am looking forward to learning new things. And I cannot imagine myself being in a job where I just need to go every day without being inspired and without expecting some developments in the things that I did the day before. So for me, I think you will be happy in a field of what you study if you have the passion and the curiosity. And of course, it takes hard work for that.
I'm actually, thank you for sharing all that, Cynthia. And I'm actually very inspired by Razia's story as well. I don't know, Razia, would you be willing to share some of how you got into cancer research and the interesting journey that you took along the way?
I did my undergrad in biochemistry, and then I went and did a master's in chemical engineering, and then I took off some time, raised my kids, and came back to like I thought, I have this degree, and I want to make a contribution to the field. And so I emailed Dr. Wahl, and he was incredible, and he said, of course you can join my lab, and he gave me the opportunity to volunteer, and and I get to make a contribution every day.
Yeah, so do you want to add anything?
Oh, yes. I was waiting. Yeah, so as someone who graduated now 3 years ago from college, so I actually have two advices. So the first thing is to just don't be afraid. Go talk to your professors. They're not scary at all. They're just regular people like us, and don't be afraid to ask questions, because they'll be able to answer questions that you have. And also, secondly, I just want you guys to start exploring for anything. Cynthia mentioned, having passion and interest is very important, and to keep that up, you gotta continue exploring, and it's so fun. That's college lets you do that. So I'm really excited for you guys and your journey into college. So I support it. Enjoy. Yeah.
Yeah, and if I may add to that, scientists are not scary. They are very good mentors. My best friends are scientists, and they can be very supportive. And mentorship is something that is important in your chosen career. And here at Salk, especially, we get it from our principal investigator, Geoff Wahl, and of course, postdocs and staff scientists like Nikki here, who is always ready to explain things and suggest things. It's just inspiring to be in this lab. Are we getting any more questions?
Yeah, Josh had another one, which was, what's something you've learned in the lab that changed the way you live your life? I can actually start with that, because the way in which my research has taken me towards, again, this looking at liver damage and how essentially the health of your body can impact tumor progression. So I second guess every time I want fast food now, or sugary drinks, or anything that we now know can really contribute to tumor aggressive behaviors. And I mean, unfortunately, is a lot of luck. If you happen to maybe you go your whole life and you never get cancer, or you develop a cancer that's very treatable, or you develop a cancer that's very untreatable. And there's a lot of luck in that. But one thing we look at is also how your lifestyle and how the health of your body can actually impact tumor progression. So I did eat Church's chicken the other day. I felt that.
We have another interesting question here that says, what degrees did you guys get to enter the field of cancer research, and at what schools? Well, for me, I got my education and basic training, my BS degree, in Manila, Philippines, as a medical technologist. So I had nothing to do with research. But then I got to another country where the job that was offered to me was in plant research. And I did that for a long time. And then someone offered me a job to come to the United States. And it was a completely different kind of research, where from plant, I went to this neurobiology lab. But as the PI, my ex-PI told me, if you can extract DNA from a plant, you can extract DNA from anything,
and you should not be afraid to learn new techniques. And that's what I did. And from neurobiology, now I am here in cancer research. So that was my path, my long path. Anyone who wants to contribute to that? Nikki, perhaps?
Yeah, because I haven't shared my degree. I know Razia and Yesel mentioned their studies. So I also took a little bit of a weird path. I went to a liberal arts university. It's a tiny university in Oregon called Willamette for my undergrad. And so, being at a liberal arts university, I did get a degree in biology with a minor in chemistry, but I also got a degree in religious studies with a minor in classical studies. So I learned Hebrew. I translated the Book of Second Kings, that kind of thing. So, unrelated to science, but certainly added to what I think of as the creativity needed to be a scientist. And then the truth is I didn't do a master's. A lot of people don't know this, but to get a PhD in biomedical research, you actually don't need a master's. Instead you probably need research experience. So along the way, what as an undergrad, and then I took 2 years off to do additional research after I finished my bachelor's. Then I went straight into a PhD program. And another secret about the PhD program is you get paid. So you don't have to pay for it, and you get paid. So it's actually a really nice path in terms of avoiding, just hordes of student debt, what you can hear with other things. So.
There is another question that just came in. It says, how much does each person in the gene expression lab do their own research, and how much do they collaborate with each other? Who would like to take on that question? Well, I think as a postdoc and as a research assistant / lab technician, there will be different answers to this. So would you like to answer first, Nikki? And then maybe me, Razia, and Yazal could say something about our experience?
I don't do anything alone. I work very closely with, Cynthia, Yesel, and Razia, as well as other postdocs in the lab. So everything that I do is collaborative, and I love it. It's you're never just off on your own island working alone. I think if you acted that way, science would move a lot slower, and you'd probably fail a lot more because people have very good insights. So, Yesel and Razia, and Cynthia, they all have great contributions and ideas to the studies that they help me out on. So even though it's like, quote, I hate to even say that, my study, it's not my studies, it's our studies, and we all work together. So.
Yeah, and just to add on to Nikki, like she said, basically, it's a collaboration almost every single day. You're more than everyone's more than happy to answer any questions that we might have, that we might not have answers within our lab. Then we go and reach out to other labs around us, and it's like the purpose of what Salk Institute was made of. Collaboration every single day. It's always happening here, and it's very open to work with other people. I mean, with current situation being making it a little hard, but definitely with this like virtual system going on, we're holding meetings, and it's a very collaborative environment, and that just makes it really more awesome to be working here at Salk.
Razia?
Yes, I'd agree with both Nikki and Yesel. It's a very collaborative environment. Everyone helps one another. And, if you have a problem, there's always somebody that you can ask, and they're willing to help you. And in each of our, I mean, I see like everybody might have their own projects that they're working on, but there's always somebody that, if they run across something, they can ask for help and people will help them.
Yeah, same thing for me. Like, I'm kind of help for everyone. So whenever someone needs help with whatever technique that I could contribute to, then I'm on it. Or if I need to learn something, it's very collaborative. That person who knows how to do things will show me, and we show each other. So I think that is also a secret to having like a collaborative and cooperative lab. We have another question, which is very interesting. It says, how did the pandemic affect your current research?
So I gotta say, as somebody who Cynthia kind of keeps the ship sailing in our lab, if you know what I mean. So I think Cynthia should answer this question.
Okay, so once the pandemic started, we were down to 3 people in the lab, and 2 postdocs and myself. And I had to jump in and do the necessary, essential responsibilities in the lab. So and for a while, we were only 3 people in the lab, but yeah, we kept afloat. And then later on, it was switched to another kind of allowing more people to come in the lab. And that's when Yizol and Razia and other technicians came in. And that's when we started to get really like in our normal, like full force work in the lab. Although there are some restrictions, like, okay, there were time limits. When Nikki used to work long hours, she was cut off to until midnight. Okay. So, but that did not stop us from doing what we had to do. So, yeah, for me on my side, it was what happened. How about you, Nikki? Did that bring you down, or?
Yeah, it was extremely tough, and that mostly because what I said earlier about having an ability to predict when my day is going to be over. So when they said you have to be out by midnight, that was a challenge. But so yeah, projects were definitely slowed, which is unfortunate. But we were lucky in that we were able to continue most of our projects at least some level of partial progression.
Okay. Another question, is there a way to improve the checkpoints in the cell cycle so that there would be less uncontrolled cell division? Would you like to take that on, Nikki?
Yeah, this is a very intelligent question, whoever asked that, because whoever asked this question understands that part of the issue with tumors is that the tumor cells just continue to uncontrollably divide. So they're not responding to cues that say like, stop dividing, we're done. And there's a lot of reasons for this. Now, to improve the checkpoints in the cell cycle. This is an interesting question because it's not just one signal that says divide or not divide. There are many signals that together integrate to tell the cell whether or not to divide. So like one of the major proteins that are involved in cell cycle regulation is a protein called p53. It's actually the #1 mutated gene in all of cancers. And that's because if you disable p53, it will allow the cell to progress through cell cycles, even though there are other cell cycle checkpoints that should still halt the cell. So it's one of those things that I'm not sure we exactly know how to tell the cell to stop cycling, even because there are still signals saying don't cycle, but they'll keep going. So it's this is a difficult question that I don't think we have the answers to right now. So I appreciate that.
Thank you so much to our incredible scientists for joining us. Thank you to Nikki. Thank you, Cynthia. Thank you, Yasol. Thank you, Razia, for joining us, for giving us a little bit of a glimpse of what it looks like to work inside the Gene Expression Laboratory under the direction of Dr. Geoffrey Wahl. Thank you so much for Dr. Wahl for allowing his lab to participate and for giving us our opening program. It is wonderful to have you guys kick off the week, and thank you so much. As we saw in the beginning, cancer research is so important, not just to study in hospitals with patients, but in the lab, so we can continue to make groundbreaking discoveries to improve the medicine in the future. So thank you so much, students, for joining us. Thank you for your patience as we work through some technical issues. We look forward to having you join us later this week for other virtual lab tours. Everybody, until then, stay safe, and we look forward to eventually being able to have you on campus with us again sometime soon. Bye, everybody.
Thank you.
Bye.
Bye.
Thank you.
Thank you.